Hello friends, I am Priyanka Hire, Assistant Professor in Microbiology of Satana KAA NMS College, District Nasik. In this video lecture, we are going to talk about gram staining or its principle and procedure. Firstly, we see what is the prerequisites of this lecture. Before learning this topic, students should read what is meant by stain or dye, what is staining, what is the mechanism of staining, also how we are differentiate gram positive or gram negative microbes, also what is the principle of gram staining. Okay. Firstly, we see the session agenda of this lecture, that is which type of stain we use for gram staining and with their roles we can discuss here. Okay. Then principle and protocol of this method and how we are classify, that is gram positive bacteria or gram negative bacteria. Okay. Firstly, stain. What is meant by stain? Stain is a dye used to color the living or dead organelles. Means bacterial staining means what? Is the process of coloring of colorless bacterial structure components using stain. Okay. There are three types of stain: acidic, basic or neutral. Acidic means negatively charged acid radicals impart color in eosin, acid fusion on Congo red, also malachite green or nigrosin or Indian ink. Also the basic is positively charged basic radical combined with negatively charged particles in cytoplasm and give its color. Okay, example is crystal violet, saffronin, methylene blue, and also the third type, that is neutral. In that neutral type of stain, both positively or negatively charged imparts different color to different components. Okay. We see the staining methods. There are two types of staining methods: positively staining and negatively staining. In positively staining, where the actual cells are themselves color and appeared in a color background, or in the negative type of staining, where the cells remain clear, means uncolored, and background is colored to create a contrast to aid in the better visualization of the image. In the type of positive staining, there are two types of staining as differentiate. First is the simple staining, that is we can say monochrome staining, means in that stain in which provide color contrast but gives same color to all bacteria and cells. And another type, that is differential staining. In that differential staining, the stain which is imparts different color with different color bacteria is called differential stain. For example, in that gram staining and acid fast staining. Today we learn the gram staining. Okay. In this video lecture, we are going to talk about gram staining, or that is the principle of gram staining, procedure of gram staining, or how exactly gram staining is done. Basically, I want to tell you the process of gram staining and exactly with which principle of gram staining is possible. Okay. Here we use the four type of stain in this method, that is first is iodine, second is ethyl alcohol or acetone, third is crystal violet, and four is saffronin. Okay. The learning objective of this technique, it is to state the principle of this technique or to differentiate gram positive or gram negative organisms. Okay. Before we start the principle of gram staining, we see the history in short, that is this technique was originally developed by the scientist Sir Hans Christian Gram in 1884 when the lung tissue sections from a patient who died of pneumonia. Okay. Here we see in this slide, that is the principle of gram staining. Okay. We see in that picture, first of all we need to understand what is gram staining, why gram staining is used for, or gram staining is used I will told you that is gram staining is used for differentiate gram positive from gram negative bacteria. Friends, there are two types of bacteria uh, completely and they are named based on the way that they are staining with the help of gram staining. Okay, if they receive the particular color, they receive the pink color, they are classified as a separate type, and if they receive the violet color, they are classified the separate type. Okay, so ultimately, as a microbiologist, if we look a bacterial stain under the microscope, based on the color differentiation, we can tell which is gram positive or which is gram negative bacteria. But how exactly we can differentiate bacteria is based on gram staining. To understand that, you need to know about the mechanism of gram staining. First, we know about the mechanism of gram staining. In the process of gram staining, we need a few things. Now, what are the things that are needs to gram staining? Let me give you the simple idea. We need a primary stain. Okay, there are two type of stain of secondary stain. That two stain we actually need. One is the primary stain and second is counter stain. Apart from this two stain, we need a mordant, which also known as fixer dye. Okay, fixer dye of the stain, which we have to use, and we also need the decolorizing agent. So these are the four components that we are need to gram staining procedure. That is primary strain and secondary strain. For apart from that, mordant and decolorizing agent. Now let's move on to the process here. Okay, so let's assume that dealing with the chunk of bacteria, we don't know whether the bacteria are gram positive or gram negative. We see in this picture or in this diagram, that is the principle of staining technique. How exactly staining procedure differs in both of them. So let at the beginning both of them are while in the picture I don't have any kind of stain them, no color at all. Okay, so the first step is fixation. Here we see the first step is gram positive or gram negative bacteria, both are differentiate, and the first step is fixation, where we put we put the bacterial culture in the slide, glass slide, and we heat fix it. Okay, not too much heat but slightly kill the bacteria away, fix it, and paste it to the slide. They comes to the first step of the staining. The primary stain is added, and the primary stain we use here, that is crystal violet. It is also known as CV. CV means what? That is crystal violet stain. CV, as the name suggests, the color is blue color, means we can say the purple color or violet color. Okay, so that the color is provided to both of them, so both bacteria will take a crystal violet stain. Now the next step, now the bacteria usually take this stain, no problem or no issue. To retain that stain, they need to have any accessory complex, that is known as a mordant, which help in the fixation of dye. Okay, as a mordant, we use here that iodine. We use as a iodine as a moderate, that is fix the dye. So now the iodine with crystal violet formed crystal violet iodine complex. We can also say that CVI complex means what? Crystal violet iodine complex. With the stabilize the crystal violet strain, right? And it will not allow the crystal violet to be escaped from the bacteria to outside, or hold on to this step. That is the job of mordant, means what? To hold this step. Okay, this is the job of mordant. That is part is done. Now the third step, we see the third step. First is fixation, second is crystal violet we can add, and iodine treatment after that. Now this is the third step, that is decolorization. It is very important and crucial step in gram staining to decolorize the bacteria. How did decolorize it? With the help of decolorizing agent, that is alcohol or acetone. We decolorize that bacteria with the help of what? Acetone or alcohol. Okay, both of them can act as a decolorizer. So it will alcohol or acetone is used, which is the crystal violet iodine complex. Now you may ask me a question. We gave iodine so that the CV stain, that is crystal violet stain, is retained, right? So after using and treating them with alcohol or acetone, how come they release crystal violet or acetone complex? The answer to it, that there lies the trick between gram positive and gram negative. If you study their cell wall structure, is cell wall of gram positive are much thicker. We see here, see in this figure, the gram positive bacteria cell wall is very thick as compared to gram negative bacteria cell wall is very thin. But in case of gram negative, cell wall of or peptidoglycan layer is very thin in the cell wall of bacteria. It is made up of peptidoglycan. So what happened here is that gram positive cell wall is very thick or gram negative cell wall is very thin of peptidoglycan layer. So CVI complex is written here. We see so CVI complex will retain by a gram positive bacteria but cannot be retained by gram negative bacteria. So gram negative bacteria has very thin peptidoglycan layer, will release the crystal violet. So after decolorizing, we do washing stage. You know, the washing stage is always involved which are not written here, means procedure is follow like that. I will show you with the help of flowchart. That is firstly we prepared or heat fix the smear of bacterial culture, then after cover the smear with crystal violet, then rinse it distilled water, means after CV we wash, after treat of iodine we wash, after every treatment we wash it to every stage. Okay, wash with water. So here these are the steps involved in gram staining. Okay, so here when we wash CVI complex from gram negative bacteria, they will wash away. So after this stage, gram positive bacteria still hold on the violet or purple color, and on the other hand the gram negative bacteria will not hold any color, they colorless right now. They finally become the counter stain or secondary stain. The last stage is what? Safranin, counter stain. The color of safranin is pink or to red. So if the put a safranin as a secondary stain in our counter stain, so who will absorb the safranin in one without the color? That is gram negative bacteria, because gram positive bacteria had already the color, so they will not take it. And in case of gram negative bacteria, they takes place the color of safranin. If showing pink color, so at the end we can easily differentiate violet color is gram positive while pink color is gram negative. Here we can see in the picture, see that what is the difference between, what is our observation? Gram negative bacteria show pink color and gram positive bacteria show which? Violet color. So at the end we can easily differentiate that violet color is gram positive and pink color is gram negative. So whole the process is done. So see how exactly look like gram positive or gram negative bacteria under microscope. We will check it like that. We will see in that figure. Now gram staining is one of the crucial staining process that we will have to use differentiate gram positive or gram negative bacteria. Okay, based on their cell wall, these are the difference between gram positive bacteria or gram negative cell wall, also the peptidoglycan layer, thickness, also lipopolysaccharide, lipid content, teichoic acid, or examples of gram positive bacteria, that is bacillus clostridium, also the gram negative bacteria, that is enterobacter or acetobacter. Means here we learn, here we observe. After performing the staining procedure, the scientist observed the culture color under the microscope. The bacterial culture appears purple. It is said that give positive gram positive bacteria, and the bacterial culture appears pink to red. It is gram negative bacteria. The results are due to bacteria cell wall. The structure is different. Okay, that's here we can conclude, or based on their cell wall peptidoglycan structure. Okay, that is idea of gram staining. Friends, I believe you have clear idea about gram staining. If you like this video, please like it and share it and subscribe it to this channel to get more videos like that in the future. uh, it is a reflection point, that is I will give you the question, that is the diisocyanate of methylene blue belongs to which group, and the correct answer is option C. Also I will check out the knowledge, and for it the question is, what is the correct order of staining reagent in gram staining? Means our first step of gram staining. Then after I will give the assignment to the student, which of the following statement is incorrect. Also I will give the student assignment, what is meant by stain. Also write down the principle of gram staining, write down the difference between of gram positive and gram negative cell wall. Also fourth question is, what are the different stain is used in gram staining? Friends, if you like my video, then please like it and share it and subscribe it. Thank you so much.